Right, well, hi everybody. Right, today's session is all about paper 2 required practicals. We're following the AQA syllabus. So in total there's 4, but one of them is there's 2 methods to it. So they've elaborated. The first 3 I'm going to talk about are just for combined science, and then the last 2 are for separates. Okay, so there will be a Q&A pause after we finish the combined science ones, and then I'll get on with the separates. And obviously combined science students are welcome to stop and leave then. So that is all fine. I'll say, well, we'll start with, actually where's the one with my pen? There we go. Right, so we're going to start with the reaction time required practical. I'm just going to say that every single PowerPoint or every single practical has a video linked to it, so you can actually watch it or watch, a teacher doing the practical. So that I highly recommend, because it's much better than looking at a piece of paper and thinking you know how it works. So definitely watch the videos. So this reaction time one is doing the homeostasis and the nervous system. So there is a little section down here, just adding a bit of knowledge to the practical, and it's basically we're measuring the time it takes for our eyes to notice and for the message to go to our brain and down to our arms to catch the ruler. So that is actually what we're measuring, and obviously that relies on the reflex pathway. And you have to remember the order of the neurons. So our eyes are our sensory cell, basically our sensor. It then goes through our sensory neuron. Now, we're quite lucky because our eyes are very close to the brain, which helps a lot in our speed. Within the brain, it then goes straight down to the motor neuron, to our muscles that then contract, okay? The resulting or the effector is either a muscle or a gland, okay? Now, this image here is typically when your sensory organ are your hands or things like that. So they have to travel up towards your central nervous system, your spine, and then go up to your brain and then back down again. Okay, this requires it takes a little bit longer, but what you've also got when it is involuntary, you have the relay neuron as well. So that's the one right in the middle, and that's in your central nervous system. Okay, so it is a pathway where the signal can boycott, basically go into the brain, because what your body wants is a quick reaction to avoid getting hurt and causing damage to the body. Okay, so the relay neuron is the reflex arc that will help with involuntary action. So you will still get a signal that goes up to your brain to tell you that you're in pain and things like that. So, for example, when you stub your toe, you know you've stubbed your toe, you instantly flinch back. That's your muscles contracting and moving your leg away. But then you pause and you wait for the pain, because you know the pain is coming, and that's because the signal's gone up to your brain, and then you know you've hurt yourself. I think I actually broke my toe the other week and it still hurts, so that's all good fun. But that is the difference. That's where the relay neuron comes in, and that's actually what's happening. But for this is a voluntary one, so it is literally going from your sensory, your eyes, sensory neuron, brain, down to your arms, muscles contract, done. Okay, so describe these PowerPoints here. I found these are really good because they literally go through typical questions like that get asked for this or these practicals. So all I've done is added a little bit extra to the slides as well, just things that I think yeah, they might also ask that. So we'll go through typical sort of questions, make sure you fully understand. Let me get my pen working. Pen. Describe what is meant by the term reaction time, and that's just what I've been talking about, but you can summarize it in a much quicker way than me waffling on. The length of time taken for a person or system to respond to a given stimulus. So again, we're just using some nice keywords. They're scientific words, stimulus. It just helps. But putting those biological terms in, it does help. Outline why reflexes are important. This often gets asked. They are fast and rapid. And what you do, homeostasis is all about keeping you constant in the right environment, internally and externally, depending on internal and external environment, sorry. And also to avoid harm and damage. That is what this is all about. Yeah, obviously you're going to be able to label all those different parts there. You need to know your neurons and the order it all goes on and everything, and your different what they look like, the 3 different neurons and what they look like, where the nucleus is, etc. Okay, how do you know the diagram below is showing a non-conscious response? This is what I've said before. The brain is not involved. The brain does get message, but a little bit later. Okay, so when you see the relay neuron or anything like this, you know that is involuntary. Okay. Right, getting down to the nitty-gritty practical bit. There are key points about this practical that are absolutely essential that always seem to come up. But here we go. Below is an experiment 2 students performed on comparing reaction time. So student A sat with his elbow placed on the table. Very important there. You can't have your hand hovering. It needs to be resting on the table. Student B held the ruler with his 4 fingers and thumb and placed the ruler at 0 centimeters at the student's thumb. Now, there's a few additional bits I would like to add here. 1, use your non-dominant arm. Okay, just helps. And second, you can also mention, because of your control variables, it is best to say have your thumb and 4 fingers as far away as possible from each other each time, because you could at one point have them like this, another point like this, another time like this. Each time they need to be as far away as possible, and then that avoids any inaccurate results, because even if you like just a little bit squeezed in like that, it's going to be a quicker response overall. So again, it's all to do with the control variables, making sure you know where you could go wrong basically as well. So student B dropped the ruler, student A caught the ruler. So yeah, record the distance and use a conversion chart to note the reaction time. There, let me just make sure, yeah. When you are recording the distance, always record it from the same point. So if we look at this diagram here, I would read that from the top of the thumb. Okay, so you would always, when you catch it, always read the ruler from the same place of the hand, where the hand's caught it. Okay, and then obviously repeat those steps 3 or more times to get an average, and then you swap roles basically. Okay, so that is the basic method, but there are a few extra bits as well that you must also remember. So this was measure from same point on hand, or finger or thumb, whatever it is, just make sure it is consistent. Okay. We still calculate the mean. Yeah, no, I missed it. That's fine. So with this practical, you need to know independent, dependent, and your control variables. Here, your independent, that's what you're changing, is the student. Dependent is your reaction time. You're measuring the distance, but then you're using the table to calculate the reaction time, which is over here, and you'll always be given that reaction sheet, so don't worry about that. Control variables. Make sure it's the same ruler each time, because they could vary in mass, and then that'd be physics involved, like dropping it. Anyway, same hand or arm being used, no distractions, arm on table each time, and put the non-dominant arm as well. And then remember that fingers apart as well. They're the critical bits then. I think that is about right. So if I move myself over there, that's fine. You'll get a table. You calculate the average, be filling in, then your reaction times, and again create an average. And that's about it. So I love this practical. It's so much fun, because what they could go on and ask as well is what factors could affect your average time as well. So we've done it where I don't know if you have, but we've had blindfolds, we've had someone drinking a cup of tea or coffee as well beforehand, or full fat Coke, having listened to music, and then you can compare then what the results are. Is it slower, faster, or whatever? So always have that in mind, that they could actually twist this practical just a little bit and incorporate that sort of factor to it. So it says, describe how the results from the student's investigation. Well, that sentence doesn't make sense. So it just says describe. I think we've crossed that out. The results from the student's investigation. With your answer to this, don't just go A is faster. Write it out in a full sentence. So student A had the fastest reaction time at, written actually the value, while student B reaction time is 0.02 seconds slower at 0.13. That is a really professional answer. You would get full marks for that. So just be aware of how you're writing and what the content of your answer is. Suggest how students could improve. So again, we normally do about 10 reps each for this one. So yeah, more reps. You could compare more students, high resolution measurements. So your ruler here, it's only to 0.5. You could go down to the nitty-gritty millimeters. Okay, so your resolution, using some sort of computer to measure. Okay, I don't know what, or could be you could film it, go slow-mo. I don't know. There's other computerized ways of doing this. Okay, what was the resolution of the ruler used in the investigation? I would disagree. I'd say that was 0.5 centimeters resolution, because you got the values in between there. And why is using a computer a better method? It's faster, more results, and a higher resolution. So you'll probably get to 2 decimal places and a lot more professional. Okay. How to improve the experiment. There we go. Use aided. Yeah, let's say at least 10 times. Yeah, so that's my opinion. So my dog has just come in. Sorry. Okay, that's your first practical and brainstorming that. So the next one for combined science is also separates as well, don't forget. Let me just move my screen. This is all about ecology. So this is actually the last module in AQA. So if you're year 9 or year 10, you might be doing that this summer or this term, because it's an outside one and you can go out into your school field and do it. That's what we normally do. They've included here, which is quite nice, is the difference between, sorry, my pen's just gone. Where's it gone? There it is, it's up there. Here it is, the biotic and abiotic. So biotic are factors that are living, okay, whereas abiotic is non-living. So for example, the sunshine, this could be competition from other animals and stuff. So know the difference between biotic and abiotic factors and how they influence the ecology of species and plants and all sorts. Okay, so here are some examples here. With this practical, what you're doing, what they're trying to get you to appreciate and understand is sort of a counting method. We as scientists, we need to count things, we need to assess things, we need to be able to measure how things change. My whole PhD was on ecology side of things as well. We needed to count things, and not necessarily plants, it was dung beetles, but you know, same thing really. But we had to put traps down, and we had to think about where we put those and make sure they were random. Okay, then you get an appreciation of what is actually within that area without having to go and count every single specimen that you are interested in. Okay, so quadrats are when you have a field and you're looking at maybe one type of plant, for example. You need to work out the area of that field and then calculate about 10% of that area. And that is how many quadrats you have to place in a random arrangement. Now, how you randomly do that, you can take a photo of the field and create little grids across it. And you can number those grids like a little like a map type thing, like a grid reference. And then you can use Excel to randomly generate numbers, and then that's where you're going to place your quadrats. It has to be random. Okay. You can then, what we've got here, state the name of the equipment, that's all fine. Yeah, so this is what I'm up to here. Random number generator, or choose number out of a hat after splitting the field or space up randomly. So yeah, if you're out in the field and you have no access to a computer, there are other means and ways of doing this, but with any answer, you must make sure that you comment that is a random arrangement. You don't just stand there in one corner throwing quadrats and not moving around. It has to be properly and officially done. Okay. Yes, so then with each quadrat, you then need to count however many daisies there are. Just remember, if there's a daisy sort of half over, you still count that as one. Okay, if it's more than half over, then you don't count that one. That's what I normally do anyway. Okay, that's another issue. Calculate, and then it's a bit mathematics. Basically, you have to estimate the population size. So to do that, you need to work out the area covered by the quadrats, the number of daisies in those quadrats, and then calculate the area of the field. And then you follow this, where's my pen? This equation here. So you have the total area. In this case, it was 100 meters squared. Area sampled, it's only a small percentage of that, but then that's 15.75. Be careful and make sure you measure it accurately. As this, in this case, it's 1.5 meters by 1.5. Normally it's 1 by 1, but you know, they like to be awkward, I'm sure. Number of daisies is how many you've counted for the whole lot. You've got you do actually have an average as well, but we don't need that in this one. And then you just work out. So this is your estimated number of daisies for the whole field. As I said here, count the actual number of daisies. If you actually took the time and counted all the daisies, then they counted 159. Now that is obviously different to that, and this is something that they could also ask. Why are they different? What the pros and cons of doing an estimate compared to actually going around counting them all? And all you have to say simply is that counting absolutely every single one of them will take far too long. It could take a long time. Okay. Yes, it is more accurate because you're actually counting them all, but it's the time factor. Whereas an estimate, you get a general idea of how many there are. Okay. So that's the key thing you have to remember. Oh yeah, and it's a lot quicker, obviously. Okay, and then just down here, there's another example with different size quadrats, different size field, just another walkthrough example there. Now, the other ecology practical is to do, oh, hang on, I've got another one, hang on. Okay, yes, so I did a bit extra ones. So control, independent, dependent variable. So your controls in this one, yeah, as I said before, you only count one particular plant. You can't count more. Well, you can, but have a separate table perhaps. But if you're looking at just one plant, then just keep it to the one plant. One type of quadrat, do not use different size ones. Number of throws or placements, that has to be around 10%. Okay. Independent is the area where the quadrat is thrown. So that is what's changing. What you're measuring is the number of plants in that quadrant. Okay. The other ecology one are transects. Now this is the same purpose is to measure the number of plants, but this time we use like a line. So it could be that you measure it from like, I've seen questions where it's from the sea, the beach, all the way through to the fields at the back. So it's going along that line, going through different ecological areas like fields, bushes, beach, things like that, and seeing how the number of plants, one particular plant, changes. Or it could be looking at all the different plants along that and how they're changing, to get an idea of what's there basically, and also where those particular plants favour. Like with the other one, the quadrat, there could be an area of the field where it's completely bogged, could be an area where there's loads of trees, so it's shaded. So you want to, where the quadrat is, that you're including areas like that, but you're not really assessing how many plants are under that tree or exposed to in the open in the middle. This one kind of, it will tell you how many plants are under the tree, how many plants are in the bog area, for example, so you can actually see how the numbers change in different environments. So that's one thing. So how a transect works, as you can see here, you must have a line, and it be with a tape measure, something like that. And you still use quadrats, but you place them every, if there's normally a meter square quadrat, place it at 0 meters, and then you move 2 meters and you place it again, count them 2 meters on, place again, count, and so on. And then you keep a tally of how many plants there are at each quadrat along that transect. And how different, light, wasn't it? So suggest there's no daisies found under the tree. This is where your biotic, abiotic factors come in. You need to link in your photosynthesis for plants. Also suggest why there was no daisies found at 12 meters, nothing found. These are just some sort of factors that will affect plant growth. There could be, could have been a path, wrong pH, poisoning, maybe you didn't count it properly, or an animal's come along and eaten the plants, just particularly there, I don't know. But these are answers that you could use. Okay, exactly the same again. Estimate population size, you can do exactly the same equation, total area over area sampled, times by number of days counted. And again, and then explain why the results from the student's investigation can be described as inaccurate. So what we want here, the transect or quadrat method is an estimation of the total. The best method acquired results will be counting the actual, again, it's just counting the actual number of daisies is the most accurate one. But obviously, same answers before. Time factor is an issue. You need to be able to do this quickly and effectively. You haven't got all day, basically. How to improve, again, very similar. What normally happens with transects, you'll do one and then move it along a bit. You could actually do the whole field every 5 meters, for example, and then cover the whole field that way. Depends how big the field is, depends how long you want to be there, but doing more transects over the field, that will increase your accuracy and the number of daisies. Let me just check. Oh, my Q&A. Bear with whilst I just pause so that you can give me any questions. Bear with. Right, part 2. So these 2 required practicals are for separate sciences only. If you are combined, you don't need to listen to these. Okay. Right, the first one, you do this, year 11, I think. It's all decay, I think it is year 11. Yeah, you are measuring, yes, when you do microbes and things like that. So you're measuring how quickly things decay, looking specifically at milk. And this links in your food practical as well, your knowledge about enzymes, as well as that fats break down into glycerol fatty acids. So it's all based on the fact that milk is an alkali. Okay, and in here we do add a little bit more sodium carbonate to make it more alkaline, more obvious. And then when something is being decayed, it is being broken down. So decay, as it says here, it's the process of rotting or decomposition. Okay. Why is milk used in this investigation? Because when it breaks down, you get fatty acid. So milk was alkali, now it's producing acids. It's going to be, the pH will change, and that is what you're going to be measuring. So depending on how much is broken down, that will be determined by the pH. You can measure it using that. So for this, the dependent variable is the color change of the universal indicator. Okay. The independent variable in this one here is your temperature. So you're going to measure the rate of decay at different temperatures. Control variables, yes is what you should be saying. Whenever there's a practical, you must think, right, volumes, concentrations, time, things like that. Yeah, individuals looking at the color, that, you know, that's subjective, so make sure it's the same person comparing the colors and using the universal indicator sheet. But yeah, those are your common, very common and always included control variables. So with this, yeah, lipase is used in this investigation as it breaks down lipids into fatty acids and glycerol. What else was there? This was it. You are again dealing with enzymes. Enzymes are affected not only by temperature but by pH. So whenever you're doing an experiment like this with enzymes, one of your control factors must be your temperature. Okay, and yeah, pH is changing. So you've got to use a water bath. In this case, your temperature must stay the same, and then you're measuring the pH. So you stick it in a water bath, and you can have different water baths going at the same time. That's absolutely fine. But you just keep them in there, and that will make sure that that temperature is maintained. Always remember, enzymes are affected by both temperature and pH. Both those factors have to be thought of and considered when doing any experiment on them. Okay. Once the lipase, remember with A's, anything A's, that's your enzyme. Okay. Added to the milk, the cresol red solution, there was a color change. Explain it, and that's just again, as we said before, the lipase breaks down into fatty acid and glycerol, creating a more acidic environment, and then it changes from red to a yellow in acidic conditions. I can spit it out, that'd be good. Yeah, things like this, they'll ask you to calculate the mean. Again, you need to repeat it multiple times to create that average. It makes it more accurate. Describe the results after 40 degrees. This links in again with your enzyme work. Again, decay, the enzyme lipase started to decay and could not break down the fat as quickly. So what is missing here? Always denatures, think about your active site, things like that. Those sort of words always involved and linked very much so with enzymes. As with any enzyme graph, it's going to be a different shape. It's not going to be a line of best fit. Okay, it's never going to be like that. Okay, always remember, enzymes have a curve. I'm just going to delete that. Bear with. Erase. Donk. So when you're drawing a line of best fit, it just alarm bells. You've got to remember. Oh God, here we go. I'm just going to try and draw this. Oh, that's not so bad. That's quite good. Remember, your line of best fit must be curved. Remember words like this, optimum. So the time taken for the color change used to be the quickest, around 30 degrees centigrade. That's what the optimum temperature for what the enzyme. Okay. Obviously talking about denaturing here. That's why it slows down. Temperature, it's all your kinetic frequency of particles hitting each other and things like that. It's a bit of chemistry and so physics there. So being able to describe and explain the graph is also important. Accuracy. Now, sometimes with separates, they will ask, well, yeah, 30, it could be optimum, but actually they might ask, how to make it more precise? And when they ask a question like that, it's normally right at the end of a long question. How do you make it more precise or accurate? You can say you'll do temperatures at maybe 25, 30, and 35. So you increase the intervals, but in a smaller range to try and figure out exactly where that optimum temperature is. Okay, so be aware of that. That's all they're asking for. What conclusions can be made from this investigation? Can we use high temperature and denature? Yeah, basically just what I've said in there. You'll be asked to describe and explain the results. Okay, but that's the principle. Again, there's a little video there for you to watch. Extra little bits, problems with this experiment. Accuracy in measuring, that's important. You must have accurate volumes, less than 10 centimeters cubed. Maintain different temperatures of the milk in water baths. Okay, make sure that stays. Make sure you calibrate your water bath. Have a thermometer in there to make sure it's always at the right temperature. Measuring the pH accurate. You can use a digital pH meter for that as well. How to improve it? Use a pipette to measure out your solutions. Use water baths. That's what we're saying. So yeah, these are other little factors that you can include, but that's what we've already talked about those as well. Was there anything else? Yes. Conditions for decay and risk factors. I've just put this in a nice little summary. So condition, temperature, amount of oxygen, amount of water. These three factors increase decay. Okay, I've just gone through with the student a question about Mount Vesuvius and why have all the people there been preserved so beautifully. And it's these three factors, basically. The Mount Vesuvius erupted and covered that town so quickly with very, very hot ash that it preserved them all as they were when it hit, basically, because the temperature was so high, it killed all any bacteria that would decay it. There was no oxygen as well, so bacteria need oxygen, and the ash just, yeah, there's no water, so therefore that's it, they can't. So that's why they were preserved so well. Risk factors, there's risk factors, I'm like, oof. But anyway, be careful things going in eyes, test tube, glass, smash, cut yourself, burns and scalds from the water bath. You're dealing with different temperatures. Please do not drink the decaying milk. That's just disgusting. Yeah, you could have allergies to milk, so please be careful. Don't want to wear gloves. Yeah, things like that. So I've just put a range of ideas there for you. Okay. The last one, the last practical for paper 2 is about plants. So this is all about how plants grow and how they need light, etc. That you're just measuring how they are affected by the different degrees of light. Okay. Explain why light is important. Again, links in with your bioenergetics and your photosynthesis module. Need to know, and definitely include conversations about chlorophyll, glucose, photosynthesis. We need glucose for growth and repair. Well, growth and repair. Well, yeah, glucose for plants to grow, respiration, cell walls, amino acids, things like that. Without that, it will have stunted growth, and it, you know, remember that without minerals, etc. Okay, and diseases. So again, it's those key words all linked together, could be used in this practical, diseases of plants, photosynthesis, etc. So it's, yeah, so that word association again. Equipment, you got your petri dishes, cotton wool, you need your cress seeds, things like that. Ruler, you need maybe a protractor as well to measure degrees of different angles. If with this practical, it could be that they're not just looking at how you're positioned with light, but also maybe the angle that the seedlings grow towards the light. So lots of different factors there. They could also talk about auxin and different hormones involved with growth and movement of the plant as well. So be aware of those as well. What is the dependent? That's what you're measuring. So it could be the curve or the height. And the independent in this particular example is the amount of light. Okay, so here you're going to plant it in or place it full sunlight, heat of seed in no sunlight, so in a cupboard, and in partial sunlight. So it might be a box or something like that that you put them in. What variables should be controlled? Always, yeah, number of seeds, amount of water you put in the petri dish and the cotton wool, number of days you measure for, or when you measure, measuring technique. So same person again, same, doesn't matter about the ruler, but yeah, make sure you know it's the same person and know what angle to where the base of the plant of the stem is, and then the angle from that. So use a protractor. No sunlight placing. Yeah, it says germination, development of a plant from a seed. Oh, sorry, my bar's in the way. Hold on. Development of a plant from a seed or spore. So that they could link in. Well, what does germination mean? It is, yeah, from when it turns from a seed and it starts to sprout, basically. And then they might ask you to plot the results. So obviously something in full sunlight will grow a lot quicker, and then you'll have to link in the reason why over there, because you've got more sunlight, basically. Think about your limiting factors for photosynthesis. Okay, explain the results in the investigation. So that's just, yeah, what I've said, more sunlight, more photosynthesis, basically. There was a bit more on this as well. So some of the practicals, the hormone or the plant grown one, they'll investigate different tropisms. Okay, so I thought I'd add these here: phototropism, gravitropism, osmotropism. So look at the words. Photo, we know that's light because photosynthesis. Gravi is gravitational. Osmosis, all to do with water. Okay, so know about your auxin as well and how that affects growth as well, that also gets affected by the amount of sunlight as well. So know about auxin. Variables, yeah, so independent, that's what you're changing. Distance or angle between plant and light source. Dependent is what you're measuring. Controls, as we said in the other slide, number of seeds, how much water, yeah, the same seeds, similar sized hole in each box. Some people put them in boxes. Type of plant, type of brightness or light source. So beware of that. If you're doing it based on sunlight and you're doing it over a few days, that amount of sunlight will change. So it's best to keep it in if you're doing the full sunlight or even partial, covered up with a box, etc. Put it in a room specifically with a UV lamp or something like that. So then you don't have to worry about the different brightness each day. Okay, because your different brightnesses might affect just one of those boxes if it was one of those seeds if it's in the sunlight, and the box one might be somewhere else. So again, a factor to consider. How to improve the experiment: you can take pictures, and then using the pictures you can draw on the picture and work out the angle. Regular intervals for measurements and for longer periods of time. Use a greater number of seeds to calculate average and reproduce results. Okay, so that's your growth, your plant growth practical. So they are the 4/5 practicals for paper 2. I will pause now. Where's my bar? And then I can answer any